Hello, everyone. Thanks for joining us today. My name is Tegan Clary, and I'm the Vice President of Marketing at Unchained Labs. I'll be moderating today's seminar, and I'm very glad you decided to join us today. Today, I have Chie Funadagawa, our US West Coast Analytical Application Scientist with me today. Hey, Chie, good morning. How are you?
Good morning. I'm good. How are you, Tegan?
I'm doing fantastic, and I'm really looking forward to your presentation on this cool topic today. Antibody drug conjugates, or ADCs, can be tricky to characterize, and I think people will find it very interesting how the Homebrew tool on Stunner and Lunatic allows for customization of some applications specific to these types of molecules. So I'm looking forward to you presenting on this.
Thanks.
Some of you may have also seen that last week we launched new standards and validation tools on Big Lunatic and Stunner instruments. Chie will touch on those new developments as well towards the end of our presentation. We will have a Q&A session at the end of the presentation. So to ask questions, all you have to do is go down onto the bottom of your Zoom navigation bar. There's a little Q&A button. Click that, type your question in, and we'll try to get to as many of those as we can. Okay, Chie, are you ready to get started?
I'm ready.
All right, well then let's go and you can take it away.
All right, perfect. Thank you, Tegan, for that introduction. So as Tegan mentioned to you, let me make sure my PowerPoint slide presenter works. Okay, so today I'm going to be sharing to you about creating custom applications for antibody drug conjugates using the Stunner and Lunatic Homebrew application. So, antibody drug conjugates, or ADCs, essentially have a covalently bound small molecule toxin that's bound to the antibody. And essentially for a lot of cancer treatments, it's been able to target specific antigens and tumors. And it's been a really successful method in order to really target specific antibodies, I would say, or specific antigens and specific cells. And it's really increased the safety and efficacy, and has really attracted a lot of just attention in the past decade. More recent research has really consisted of optimizing the links, coming up with new payloads, increasing the payload, and really developing a new generation of ADCs in that sense. The drug-antibody-ratio, or the DAR for ADCs, has been really crucial to understanding the therapeutic efficacy and pharmacokinetics of ADCs. But the reality of it is that a lot of these methods that are available right now tend to be time consuming, or consumes a lot of sample, or it needs a lot of extra prep time in order to really have an understanding of the DAR. Or in many cases also, additional reagents are required in order to be able to determine the drug-antibody ratio. Out of these methods, the UV-Vis absorbance spectroscopy is the most straightforward and probably the most simple process, where you can just measure the absorption spectrum. But even with that, the number of samples that you can run can be limited. And also in many other cases, you have to measure the absorbance and then take it into a separate piece of software, and then calculate the DAR from there. This still takes time. And as we all know, we're always under the pressure against time, and time can be overwhelming. This is where I would really like to introduce to you the Stunner and Lunatic. The Stunner is, the Lunatic is essentially a 96 sample measurement for UV-Vis. It takes about 10 minutes or so, and it's in a plate format. The Stunner is essentially everything the Lunatic has in the sense of the UV-Vis system, but it also has the dynamic light scattering system inside of it. And it can read both UV-Vis, so the absorption spectrum, and the dynamic light scattering in about an hour, again in that plate format. I'd like to first focus on the Lunatic and introduce to you the Lunatic. One of the key really unique features of the Lunatic is the microfluidic plates. So how that specifically works is essentially you can see that the green circle that you see in step 1 is where you load the samples. It's just 2 microliters of sample that is required, and then through capillary action it goes into the squiggly parts that you can see in step 2, and it stays in there. From there, essentially inside of the instrument, it gets drawn into the cuvettes, which is in step 3 with the two little diamond-shaped areas. We have that in the plate format for our big lunatic. We do have a smaller instrument of the lunatic, which is in the smaller chip format. The advantage of this is really that with just the same small 2 microliter volume requirement, you can essentially load into this 96 well plate format. It's the same spacing as a regular 96 well plate, but also because of the microfluidics, and once it goes into our little reservoir area, which is the squiggly part, we see little to no evaporation up to 2 hours. So that is essentially liquid wise. We don't see anything leaving. We can also avoid any cross-contamination that may occur. And essentially, we also have fixed path lengths for our UV-Vis measurements. As many of you may remember from Beer's law, the path length is crucial to really understanding the concentration because that's how the absorbance is determined. Additionally, because of the microfluidic plates and wells also, you can really measure up to high concentrations with no dilutions. This system is also automation ready, so we are able to integrate them into a lot of automation systems. To give you a little bit more of a specific idea of what the concentration range is and the specific path lengths that we use, we have 2 types of plates. We have a regular lunatic plate to the left, and then the high lunatic plate, which you can see to the right. The key difference really is that the regular lunatic plate has 1 cuvette per sample, and then the high lunatic plate has 2. So the advantage of that really is with the high lunatic plate, the 2 path lengths is a 0.1 millimeter path length and a 0.7 millimeter path length. It's still the same 2 microliter volume requirement for both types of plates. But because of that really small 0.2 millimeter path length, we're able to go as high as 200 mg per mil for a sample like IgG. Specifically, what that means in absorbance range is about a 275 OD absorbance range, and that's how high we can go. For some of our other users, such as in our nucleic acids area or the molecular biology area, they never really have to go that high of a concentration. So that's really why we have the regular lunatic plate, which has a little bit lower dynamic range. The Lunatic also has really easy to use software. We have 2 pieces of software that come with the instrument, the Lunatic client and the Lunatic analysis. So for the big Lunatic client, it's essentially what connects to the instrument where you can run the experiments. The analysis is where you can look at your data, export, and make reports and so on. The Lunatic measures the full absorbance spectrum between 230 nanometers and 250 nanometers. So every measurement you make for the absorbance on the Lunatic is measuring that full range. As you can imagine, with just the need to understand the amount that you have, the purity, how stable it is, cultures, and so on, that this instrument is really able to be integrated in multiple steps in your workflow. But really the advantage in a lot of our protein customers, and specifically since I'm talking about the antibody drug conjugates, with antibodies being able to go to as high as 200 mg per mil whenever needed. We can essentially measure that high with very good precision with no dilutions. Here you can see how there's a very linear range of the absorbance and the targeted concentration of the antibodies. The Stunner instrument is essentially everything the Lunatic instrument has in the sense of the absorbance section of it. So it can measure the concentration with a very similar software. Actually, the software for the Lunatic and the Stunner is integrated, and it looks the same essentially. However, the key difference with the Stunner is that you can measure dynamic light scattering with it also. It has a DLS system inside. What that allows you to measure is the sizing, so the hydrodynamic diameter and the polydispersity of your samples. Additionally, you can measure the B22KD, which looks at the colloidal stability of the samples, and also the Homebrew application. Now, the Homebrew application is available on both the Lunatic and the Stunner, and it's essentially a piece of software that allows you to look at your own, create your own application. So what is the advantage of the Stunner? Well, in many places, we're always used to looking at the concentration, always checking what concentration do you have after you take it out of the fridge, after you've stored it, or in different processes. But why not get some quality information at the same time, in the sense of, are your samples already aggregated? How do they look? Is there a difference from before storage to after storage, or before a certain process and after a certain process? The Stunner can measure this at the same time with the same 2 microliters of sample. It'll measure the absorbance and that dynamic light scattering information using those same 2 microliter samples. The Stunner plate format is just one type. It is the one with the 2 cuvettes, which is equivalent to the one with the 2 cuvettes in the Lunatic, but it has the 0.1 millimeter and 0.7 millimeter path length. Again, just 2 microliters of sample, and for the absorbance range, where you're looking at the absorption spectrum, it measures that full absorbance range between 230 and 750 nanometers. For the dynamic light scattering range, so what kind of hydrodynamic diameter ranges can we measure? It's between 0.3 nanometers and 1,000 nanometers. Just to kind of give you an idea, a lot of antibodies are generally between 10 to 15 nanometers or so, depending on what you have. Sometimes a little bit smaller than 10, but that kind of gives you an idea of what the range is for hydrodynamic diameters. For the Stunner instrument, you can also just measure the absorbance. You don't have to commit to measuring a dynamic light scattering read every time, or a DLS read. If you just measure the absorbance, it's about 10 to 12 minutes per read. If you read a full 96 plate for the concentration and the DLS read, it takes about an hour. So what can you measure with DLS? As I already mentioned, you can measure the average size, so the hydrodynamic diameter of the particles, and the polydispersity of the sample. How uniform is your sample? Do you have different sized particles present, and so on? Specifically, if the particle size is more than 3x in the size, the overall volume of the sample, you can see separate peaks also appear, where you may see large aggregates as a separate peak. And really, you can kind of get a sense of if there's any aggregation or how that may change over time and at a given condition. Just as a very quick example, this is just showing 2 different protein samples that you may look at. One is very homogeneous in the blue, and it has about a 5.9 mg per ml concentration. You get that information at the same time with the same 2 microliters of sample. But you could also look at the spectrum in the green, where you can see it's a lower concentration, but you can also see clearly that there's 2 peaks that are present. There's something in the 100 nanometer range. So there's clearly some kind of large aggregates that have already started to form. This may be due to storage or some kind of process or development that you may have gone through. Specifically for ADCs, the antibody drug conjugates, this could be advantageous because you could look at the antibody by itself before you conjugated the drug, and then check it again after you've conjugated the drug and see if it's forming aggregates, if it's causing some kind of instability in the sample itself. Now for ADCs, the ultimate goal is to get the drug-antibody ratio, the DAR, with the UV-Vis absorbance measurements. So again, with the absorbance measurements, you can get this both in the Lunatic and the Stunner. Ultimately, we want the concentration of the drug and the concentration of the antibody so that you can get that ratio. Based on Beer's law, we know that you can get the concentration from the absorbance from the antibody, which most proteins usually absorb around 280 nanometers, and wherever your drug or that you've attached absorbs also. In this case, you can see about a 252 nanometer absorbance of the drug, shown with the red dotted line. The reality is that some drugs that you conjugate may overlay, like you can see in this example, with the protein concentration. So you really need to be able to kind of deconvolute that in the sense of being able to measure the absorbance at a specific wavelength and then essentially subtract out any other contributions that may be done. This is just an example of something that you could do that with. So this is essentially a chapter from a book, and you can see that it's suggesting a specific way to calculate the concentration of antibodies and drugs by subtracting out any contribution from the other species. So from the antibody, if there's any absorbance from the drug, then you can subtract that out. But when you're measuring the UV-Vis absorption spectrum on a classic instrument, what you would have to do is measure the absorbance of 2 wavelengths, export that out, and then do all of this calculation later in a different computer or so on. Sometimes when we're in the lab, we're just in a time crunch. We just want to quickly measure the absorbance, get a good idea of the DAR, and then move forward. Maybe we're ready to run an experiment, ready to run something really quickly. We don't really always have time, or maybe don't want to have to go back and do more calculations. This is where the Lunatic and Stunner's Homebrew application can really help you alleviate some of those pain points. So the Homebrew application is essentially just a click of a button on the software. This is a screenshot from one of our softwares. You click on the Homebrew application, and essentially you can create your own application. So you can click on it and you say, create a new application. Once you create a new application, you can give it the name of your choice, my special app, and then essentially say, what kind of protein type do you have? Do you have a protein, DNA, RNA, or something else? It's really just used to determine, the area to categorize the sample of the application. Because once you create this application, it's going to be there as just an application that you can select every time you run this instrument. You can also select the wavelength of interest, some visible scales. Just because you selected the wavelength of interest, that does not limit you to only seeing, only measuring that specific wavelength. You have full flexibility, and I'll show you that in a second. Also, the visible wavelength range, again, the Lunatic is going to measure that full absorbance range all the time, between 230 and 750 nanometers. It's just really so that you can focus in on the area of interest when you're looking at the picture on the software. Once you've created some of the basic informations about the application, so the name and what kind of application it is, then you can really customize it into detail about what kind of outputs you would like. In this case, you can see the lower on the list to the left, the outputs, you can see that I have 4 extinction coefficients. So I have the extinction coefficient of the antibody, the extinction coefficient of the antibody at 280 and at 252. I have also the extinction coefficient of the drug at 252, which is where the absorbance maximum is of the drug, and also the absorbance of the drug at 280, so that we can subtract out those contributions. Again, this is just basing it off of that one equation that I showed you earlier. The advantage of typing in the extinction coefficients individually is that once you have an output, that now becomes an input. And what that means is you can select it when you're creating certain equations for your output. You can always delete formulas. If you typed something in and you realize you didn't want that later, you can always delete it. And you can also specify how many digits of precision you would like to show. So here's an example of what I mean by the outputs now become inputs essentially. But you can define an output. You can see right now it's selected to the drug concentration in the output section in the left. And then you essentially type in the equation and just the calculations that you want it to do. Again, once you do this once, then the software is going to do this calculation automatically for you and essentially give you the drug concentration, the antibody concentration, and so on. Just type it in, do the calculations, and do multiplications, and then you can see in the gray box in the middle with all the little squares, you can see that the extinction coefficients are there, so you can select that and click on it so you don't have to type that in every time. It's really handy when at the end, after you've created the outputs of the drug concentration and the antibody concentration, again, just a click of a button from the previous outputs, you just say the DAR is equal to drug concentration divided by antibody concentration. It just brings in all of those calculations for you. Again, once you've created that application once and you've created the math and everything in the way you want it to be, then you can essentially see it as an output. So you can see there's the absorbance spectrum and you can see the absorbance there. And then boxed in blue, I wrote, you can see the drug-antibody ratio. So this is going to be an output every time that you just measure the absorbance spectrum of the sample. Again, let the software do the work for you so that you don't have to go in and do the math and all of that separately. The software also does have easy export and report settings also. So once you're done and you need to export everything out, it can be exported as an Excel, CSV, text file, and so on, whatever you decide. This is just showing you the export of from Excel, and essentially you can see the replicates inside of the larger Excel box. Because ultimately, when you're measuring multiple samples, you want to be able to compare it really quickly. What is the drug-antibody ratio of the different samples? You can clearly see it with the green arrow that there is a difference of the amount of drug that is present in these samples, in these ADCs. But you can really quantify that and see that number right away if this Homebrew application is already ready to go. Again, the absorption spectrum part is available for both the Big Lunatic and the Stunner, because it has the absorbance measurement section. The Stunner has additional capabilities where you can look at the dynamic light scattering portion. So you can look at that quality. You can look at the quality of the drug-antibody ratios. Is there, the quality of the ADCs with different drug-antibody ratios. In this case, you can see that there's a clear increase in the zeta average, or the hydrodynamic diameter, essentially, of the samples with additions of higher and higher DAR, or with higher and higher DAR. Additionally, the polydispersity index is what tells you in DLS how monodispersed and polydispersed your samples are. If it's below 0.1, it tends to be a very monodispersed sample. And it looks pretty good in that sense. Again, the advantage of the Stunner is to be able to provide more than one piece of information at the same time. So if you look at the the ADCs, you can check to see if there is a clear difference in the stability. Are some samples already aggregated? In this specific example, I don't have any that aggregated specifically due to the antibody drug conjugate, but in some cases you may see some samples aggregated, and that's valuable information that you can obtain. Using the Lunatic and Stunner absorbance measurements, so specifically the absorbance portion, you can really obtain incredibly precise concentrations, again, due to that microfluidic cell and having that set path length. In addition to that, with the Stunner, you also get information about the sizing and polydispersity. What is the hydrodynamic diameter? Do you have aggregates already present? Therefore, are your samples already like polydispersed, so not a uniform sample? Additionally, for ADCs, it's extremely powerful to be able to create your own applications and essentially have that flexible data analysis and have specific outputs to your needs, to your ADC, or to whatever else that you may be looking at. You can essentially make that calculation once, and now every time you select that application, it will give you those outputs that you have specified. And so with the Homebrew application, it can really take away that extra step of taking absorbance measurements and then calculating it further in a separate software. It does that all in this one software. You don't have to walk around and do additional calculations or exporting later and then calculating it later. because we know that flexibility can really be comfortable. We're also very excited to announce the Lunatic and Stunner software version 7. The Big Lunatic and Stunner qualification is accurate from the lowest concentration to the highest concentration already, but with this new version, we've even honed down further on this precision of these samples. We've decreased the percent error to 2% from the target concentration. And in this specific example, you can see that there's a dilution series of IgG from 0.2 to 200 mg per ml. This is actually done in octuplets. So there are 8 replicates that are overlaid on top of each other in this figure. So you can see the incredible precision of this instrument, and and see essentially the percent error also, really showing you the accuracy of the new Lunatic and Stunner absorbance measurements. Now you can also prove the accuracy of the Lunatic and the Stunner in the sense of using essentially 5 different separately qualified tryptophan concentrations. So you can check the actual value and make sure that the accuracy is indeed there based on these separately qualified tryptophan measurements. The reason we're using tryptophan is really because for proteins, tryptophan is indeed what tends to absorb the most in the 280 region. So we wanted to make sure it was relevant to our protein users in addition to just essentially that whole range in the UV region. You can see here again, we're overlaying in the spectrum in the top right, 16 replicates of absorbance of the tryptophan measurements. And you can see a clear and clear linear trend between the target concentration and the tryptophan concentration that was measured using the Lunatic and Stunner. For those labs that are doing manufacturing, quality assurance, quality control, or who just want to comply with international standards, the performance verification is also available on the UV-Vis system. This is available as a service from Unchained Labs, and it consists of performing 6 tests with 8 sealed 10 millimeter cuvettes that are of essentially certified references. Essentially, these cuvettes, these sealed cuvettes, will fit into this custom metal frame, which you can see in the image in the right. And essentially, these tests meet the US Pharmacopoeia and European Pharmacopoeia guidelines for UV-Vis spectrophotometers, and really ensures the suitability of UV-Vis performance verification tests on the Lunatic and the Stunner. This is essentially available to all Lunatic and Stunner instruments that and version 7 softwares that were sold after April 21st of this year. If you are interested in learning more about the performance verification or the fundamentals using the tryptophan, there are application notes and tech notes which are available at unchainedlabs.com. We also have a follow-up webinar coming up on May 14th that goes into more detail about how the Big Lunatic can be used in upstream and downstream protein labs. With that, I thank you for your attention, and I'd be happy to take any questions.
Hey, thanks, Chie, for sharing how Homebrew tools on Big Lunatic and Stunner can help people with some of these complicated molecules, right, and create some custom applications. I think that was a great overview of that. We do have some good questions already in the queue, so please don't be shy. If you do have a question, please post it in there. You can still submit it. Just a reminder, the Q&A section down at the bottom of the navigation bar in Zoom. So Chia, let's do this. Let's go ahead and dive into the first question.
Right.
So the first one here is it possible to measure various samples with various buffers using these systems?
Yeah, that's a great question. So since it isn't a 96-well plate format, I can totally understand where that question's coming from, because you may not have 96 samples of the same buffer composition. The answer is yes. With the Lunatic and Stunner software, what you can do is essentially have separate buffer blanks and essentially group them together. And you can say this buffer goes with these samples, this other buffer goes with these other samples. And so you're able to really have full control of that and make sure that you're subtracting the correct blank buffer from the correct samples.
Okay, cool, great, Gia. A couple questions just about Homebrew. First one here is, does Homebrew take DLS data as well? I think you showed quite a bit of some of the UV-Vis side of it, but can you do things with DLS in Homebrew?
So you have control over like reporting DLS or not, or certain outputs. You don't have control over the, like if you want to recalculate the DLS in your own way, it's not able to do that though.
Okay, okay. I think this is a highly relevant topic with what's going on in the world right now. Do we have any data on viruses, viral vectors, or VLPs using these instruments? I think that we can just expand that beyond Homebrew, but do we have any data we can share? And then I think, do we have any customers working with those molecules?
Yeah, so we do have people measuring this in the viral vector world, absolutely. You can imagine the advantage of it really is that with those microfluidic plates, once you load the sample, it gets inserted into the microfluidic channel, so it's not exposed to the samples, which is an advantage for viruses. So we do have some users looking at the protein concentrations of that. There's also some papers out there that you can look at to really kind of look at the titer of the samples, like for example, of the sample, like of your, using UV-Vis essentially. So we do have some users looking at that. If you are looking for specific information, I'd really encourage you to get connected with us and we can share more of that offline too.
Right. A couple of questions, Chia, that are kind of about the hardware and software. So first one here, does Stunner have temperature control, and does it do can it do a thermal ramp?
Yeah, so the answer to that is no. The Lunatic and Stunner is essentially just at whatever, like ambient temperature, essentially. So there is no thermal ramping capability.
Right, right.
So for that, we have a separate instrument for that. We have the Uncle instrument, which does have the capability to do thermal ramping and look at thermal stability.
Right. Regarding the software and networking, this question here, is the software compatible with networks, or is it constrained just to using USB? So how do people get it into their environment?
Yeah, so the kind of basics of it is the instrument comes with a laptop, and then a lot of people do connect that to their network. So we do have a lot of users that do that. Additionally, for users that are integrating this into an automation system also, you can bypass the computer and essentially connect the instrument specifically to, well, whatever network you have, but also whatever API or automation system that you may be using also.
Right. The question here about just older systems, you know, I think somebody here has a couple of older systems. Do we sell software updates to get Homebrew and the multi-blank capabilities?
So the answer is, so yes, it depends on which older system you have, I guess. So if you have some of our very, very old systems with different names, then it may not be compatible with that because there were some significant hardware changes there. But if there are more recent, like if it's called the Lunatic and it looks similar to the images I showed you today, then you can upgrade your software and get the Homebrew application. Just be aware that the verification test that I spoke about at the very end, that is only available to the newer systems though, with that 2% like accuracy, where we've made it even more accurate.
Right, right. For any participants here with an older system, my recommendation is you just reach out to us and we can tell you what things are available based on the age of the hardware and the software compatibility going forward. But reach out to us and we can help you with that.
Absolutely.
A question here on the viscosity. So is there a range of viscosity that the analysis is compatible with on these systems?
Yeah, so the viscosity limit for the instrument is really the limitation of the sample being able to be drawn through the microfluidics. So with microfluidics, obviously there is a limit. The limit is in centipoise, it's 40 centipoise is what we have right now. And you can actually go even higher with the newer systems with some new upgrades that have been done in that too. 40 centipoise in like protein concentrations, I mean, obviously we can go up to 200 mg per mil. We've measured in that range with no problems. So it's very high in that sense if you're thinking of protein concentration and viscosity.
Right. Chia, this is a question we get often, so I know you will have the answer. So the comparison, how does a measurement of aggregates on Stunner compare to measurements of aggregates using size exclusion columns? I think this is a common question.
That is a very common question. So one of the key things that we really want to emphasize is you don't want to directly compare every piece of data from SEC with DLS. They are different systems, they're looking at different things. Specifically with DLS, the size for it to be in separate peaks, so I showed you earlier, there were some pictures or images where they were showing two separate peaks. For them to be distinguished as separate peaks, they need to be 3x in size. And when I say size, I mean volume. So when you think of that in like a 3D image, it's like a 15 mer difference. So essentially what the DLS is really able to do is look at the larger aggregates, which may just go right through the SEC column. And then what you can see in the DLS is either like a broader peak, so you'll know it's polydispersed, but you won't be able to kind of distinguish what % of different, monomers, dimers, and trimers you may have. That's not really what DLS is meant for doing. One thing that I really like to kind of encourage customers or our users to do is to think of it as a screening tool. So once you can do it before SCC or after SCC and kind of decide, do you want to move forward with this sample or just do a quick measurement after it's been stored and see if there's any quality change in that sense too.
Okay, a couple of questions, Chia, for you on the Homebrew. So this one's pretty interesting. So if somebody makes a Homebrew application, right, on their own, and they catch a mistake after they actually run it, is there anything they can do with that data?
Yes, there is. So I'm actually very excited that that was asked. So the Homebrew application, one, you can always edit it. So you can always edit a Homebrew application if you don't, and you can always rename it as something else also. Once you've measured a whatever absorbance spectrum and you want to recalculate it with this new edits that you've made, you are always able to do that in the Lunatic analysis software and Stunner analysis software. So that's actually really nice in that sense where you don't have to run the samples again, you can recalculate the data in there.
Okay, great. Another question about the application. So the Homebrew applications seem to be managed in this software. Can people have multiple Homebrew applications, and can you delete them? Can you edit them? Are there things you can do after the fact? I think you mentioned part of that, but how are they kind of managed in the system?
Absolutely. So within the the Lunatic and Stunner client software, which runs the instrument, that's really where a lot of the Homebrew applications are made and edited and so on. At any point, you can easily delete them. So let's say you have a project that isn't going to be used anymore. It's just taking up space on your screen. It's quite literally a click of a button. You're able to delete those softwares and specific ones. You can also hide some. So let's say you don't want to quite delete it, but you want it to not show up on the screen. You can also hide different ones. And also at any time, like I said, you can also edit them very easily.
Great. Okay, thanks, Chia. And I think this will be the last one for the day. On these systems, what's the maintenance required on these, on Lunatic and Stunner? Are they hard to keep around in the lab, or are they super easy?
Yeah, no, so it's a great question. So for the Lunatic and Stunner, the UV-Vis portion of the instrument, every instrument comes with a calibration plate. And that plate essentially has 3 portions of the calibration. It has a pump calibration, absorbance calibration, and a position calibration. The absorbance calibration essentially just makes sure that the absorbance looks the way it should, and the pump and position is essentially because the instrument needs to go to each position of the well in order to draw your sample into the cuvette. It checks that too and calibrates it. How often often you have to do it. We say minimum, do it once a year. It doesn't hurt to do it more often. And this plate, you keep using it. You don't need to add anything to this plate. Keep rerunning it, and it takes about 5 minutes. So, or some of our users just have it part of their SOP to once, and it's very easy and straightforward. For the Stunner, because we have that dynamic light scattering portion of the optics, that one has an additional just check that you can do using a dynamic light scattering just calibration standard that we have. You samples into it, you just the essentially intensity of the DLSer.
I want to again thank any of you that joined today on demand as well. But I think with many of us still working from home, it's a great time to explore some of these new tools and ideas and look for new solutions to problems. If you would like to have a more in-depth conversation with someone from our team about Homebrew, about the Lunatic or Stunner, please do get in touch with us. I think it's very easy to connect with us just at info@unchainedlabs.com. Our team would love to connect you over a Zoom meeting. We do this all the time, and we're actually having a lot of fun just connecting with customers, talking to scientists about problems they're trying to solve. And folks like Chie and the rest of her team are ready and willing to do virtual demos. They can actually do a demo over Zoom with you if you're interested in doing that and taking a closer look at one of our solutions. We do also have quite a few other virtual seminars coming soon, as Chie mentioned. So there's a queue of those on our website at www.unchainedlabs.com. In the events section, take a look at those, quite a few. And there are many on demand already from the last, I'd say, 4 or 5 weeks. We've been doing a bunch of these. So lots of different interesting topics. So Chie, thanks again. And thank you again to all of you who have joined us today. And I hope you have a great rest of your day.
Thanks. Bye.